Good evening, everybody. Welcome to our webinar about the COVID-19 vaccine today. Today, our researcher will be Dr. Kate Broderick, who will be working in Innovo Pharmaceuticals. So they are developing new type of the DNA vaccine against COVID-19. Dr. Kate Broderick has a broad background in the product development and device development, DNA therapeutic and drug delivery field. So she received her PhD from University of Glasgow, Scotland, and conducted postdoctoral research at the University of California, San Diego. After that, she joined Innovo in 2006, where she currently leads a diverse research group focused on enhanced delivery techniques for gene-based therapeutics as the senior vice president and preclinical research and development. She is a co-inventor of multiple patents related to DNA vaccine delivery and has served as a principal investigator on grants award contracts from leading government agencies, including the Bill and Melinda Gates Foundation, the National Institute of Health, the U.S. Department of Defense, the Small Business Innovation Research Program. This totaled 56 million awards from Coalition of Epidemic Preparedness Innovations. With a keen interest in electroporation device design in the context of transdermal delivery, Dr. Broderick has helped drive the development of novel prototypes and the design of Innovo's proprietary smart device, namely selectra, which delivers the company's DNA vaccine directly into cells in the body. So welcome, Dr. Broderick. So now, before we start the presentation, we have some housekeeping rules. So all of the mic of the audience has been muted. So if you have any questions, then please type on Q&A window. So then I will deliver that question directly to Dr. Broderick. So if you have any kind of concern or question, you can also send me a message to chatting or any other means. As you see here, so we have turned off the speaker of all of the audience except panelists and then Dr. Broderick. So Dr. Broderick, welcome to Qatar Biomedical Research Institute. So we are leading institute in this Midwestern area, focusing on genetics, neurological disorders, diabetes, and cancer. So it is our honor to host you as a guest speaker for your new development and to be updated your DNA vaccine. So floor is yours.
Thank you so much, Dr. Kim, and thank you so much for the invitation to speak here today and this evening for you. It's the start of the day for me here in California. It's a real honor and a pleasure to be with you today. And I hope to spend the next hour kind of covering really what we at Innovo have been doing for the last year. And you can probably imagine it's been completely dominated by our efforts to generate a vaccine against the devastating disease that has been COVID-19. So I'm going to talk through really our activities over the last 12 months and then end up by talking through really what we are planning to do for the future, because although it might not be something that people want to think about right now, it looks like COVID-19 as a disease is here to stay. And so there has to be a lot of preparations in regards to how we deal with the future of this pandemic. So again, thank you so much for having me to speak today. It's a real honor and a pleasure. And I look forward to taking your questions at the end of the slides. So just a little bit about Innovo as a company. So we are a mid-sized US-based biotech company. Our headquarters are on the East Coast in Philadelphia, and I have the great luck to be based in sunny San Diego in California, where all of our research and manufacturing activities take place. And this is a picture of our manufacturing facility here in San Diego. We're about 350 full-time employees, and that number is rapidly growing, especially because of our efforts on COVID-19. So of course, everybody on the call this evening will be completely aware of the fantastic efforts by Pfizer, Moderna, J&J, Oxford, AstraZeneca, at really quite remarkably generating incredibly efficient and effective vaccines for COVID-19. And of course, they're being widely rolled out globally now against this pandemic. However, of course, there are 8 billion people on this planet, and really until all of those people have been vaccinated, we're not really safe from this disease. And so it's likely going to take a variety of different vaccines with different profiles that will really allow us all to be vaccinated and hopefully see the end of the devastation that's been caused by the virus SARS-CoV-2. So you're probably fairly familiar with the RNA platform from the work of Pfizer and Moderna. However, I could certainly talk about that in the question and answer session. But what our technology at Innovo is slightly different. We, instead of using RNA, we use a DNA vaccine approach. So in taking a part of the genetic sequence of the virus. So in this case, for SARS-CoV-2, we've taken the genetic sequence of the spike protein. We've taken that small piece of genetic material, we've encoded that within a DNA plasmid, which is a circular piece of DNA. We've manufactured that at high scale, and I can certainly talk at length about how we do that. It's actually an E. coli-based fermentation process. It's formulated in a very simple way, so quite different to what you see with the RNA vaccines, which have quite a complex formulation. DNA vaccines are really just the DNA, water, and a little bit of salt. And those are actually injected into the skin of the patient, not the muscle, like we probably all of us are mostly familiar with vaccines. And that really elicits the downstream immune responses that I'll talk a little bit more about as we move through the slides. So specifically, I wanted to start by really detailing 5 different characteristics of DNA vaccines that set them apart from some of the other candidate vaccines that are in development at the moment. So I'll just work my way through these, and these are going to kind of start as an introduction to the rest of the slides in the presentation. So firstly, we've really demonstrated with our COVID-19 vaccine, but really all of the DNA vaccines that we've generated over the last 20 years, that these are really very safe, very tolerable, and very easy to administer vaccines. I'll talk in more depth about our safety profile of our vaccines, but suffice to say that we really don't get those systemic side effects that are very common with RNA vaccines or viral vector vaccines. We don't really get any of those flu-like symptoms or fatigue symptoms. Really, primarily what we're seeing is some localized injection site reaction where the vaccine's delivered, so a little bit of redness in the skin of the arm. That's primarily really the most common side effect that we see. As I mentioned, it's our vaccines delivered intradermally. So instead of using quite sizable intramuscular needles, we're using something much, much shorter. And so it's a much less invasive process than standard IM injections. And it really only takes a few seconds to administer our vaccine in a very similar time frame to what you would imagine for any standard intramuscular injection. Our vaccine for COVID-19, but also for all of our infectious disease platforms, which have included Ebola, MERS, Zika, HIV, Hep C, Hep B, and a variety of others, have been shown to be very immunogenic. And that's very important, especially when we're dealing with a new virus like SARS-CoV-2 is, really. I mean, we've only known about it for a year now. And we still don't fully understand the mechanism by which we're protected by our immune responses to this virus. So creating a very broad immune response is really very key. So our vaccines give you a very strong antibody response, but also a strong T cell response. And we believe that that's going to be very important as we think about these new strains of SARS-CoV-2 that are emerging. And I'm going to touch on that a little bit later in the talk too. One key area, which I'm sure you've all read about in the media, is the stability of the available vaccines at the moment. Whilst incredibly effective, Pfizer and Moderna vaccines do require cold chain, freezers to store and transport their vaccines around the world. That's absolutely not the case with DNA vaccines. In fact, we could store our vaccines at room temperature, so anywhere between 19 and 25 degrees for 1 year without any impact on the stability. And actually, we have 1 month of stability at 37 degrees. So as we're thinking about shipping this all over the world, I'm sure in Qatar it gets very hot in the summer. You know, we don't have the restrictions needed of these ultra cold storage, these -80 freezers required to store or transport our vaccines. Plasmids DNA in general are incredibly characterizable and scalable, and I'll talk a little bit more about that as I move through my slides, but essentially you might have the best vaccine in the world, but if you can't make enough of it, it's fairly irrelevant. Whereas DNA vaccines themselves are incredibly easily manufactured. As I said, it's just a fermenter process using E. coli, and I can certainly talk a little bit more about that later on. And then last, but by no means least, as I mentioned in my opening statement, unfortunately, it looks like COVID-19 is not going to be an emerging infectious disease that's just there and then goes away again. It looks like it's more going to be a situation where it becomes endemic globally. Now that has major ramifications when we think about re-administering vaccines, so we should perhaps think about it more like a flu and influenza situation as opposed to a MERS or a SARS where it was there and then no longer. And so from that perspective, it's very important to be thinking about vaccines that can be re-administered. Certainly some of the viral vectored vaccines have a limitation on the number of times they can be given due to anti-vector immune responses. And also some of the RNA vaccines have some issues with reactogenicity, the more times that you're given those vaccines. So DNA don't have those associated side effects. We're able to re-administer DNA vaccines really unlimited amount of times with no impact on anti-vector responses or reactogenicity. So this is really just a summary slide of what we did essentially in the last 12 months. Well, very early in January, we received the genetic sequence of SARS-CoV-2 from the Chinese authorities. We immediately designed and did this in silico, of course, using a computer algorithm. The design of our COVID-19 vaccine, as I said, it's a DNA version. It's a 2-dose regime in the clinic, so you get 2 immunizations, one at day 0, one at week 4. And as I said, it's delivered into the skin. We've completed our phase 1 trial that was based in the US, and we're just about to finish up our phase 2 trial, which is also a US-based trial. However, in parallel, we also have ongoing clinical trials in South Korea and also in China. We've been working extremely hard over the last 12 months to really build up our manufacturing capabilities so that by the end of the year, we will have hundreds of millions of doses of our vaccine available. And as I just mentioned, we really are very proud of that excellent thermal stability profile that we have of our vaccine. So this is, a lot of words on this slide, but really what this just summarizes is our target product profile for our vaccine. This was generated in conjunction with the WHO guidance on what they're looking for COVID-19 vaccines. So really it just kind of compiles all of the information about our vaccine within this sort of short tabular form. You can see we have our target populations, our uses and indications, and our efficacy, safety, tolerability, and safety and distribution. So this is a really nice cartoon, I think, which helps kind of depict really our technology in an easily digestible way. So starting up there with the virus, and of course, in this case, we're talking about SARS-CoV-2, but I certainly personally have worked extensively for Ebola, Lassa fever, MERS, and HIV. So we analysed the sequence of the virus. We came up with the spike protein, which we believed would be the best area to generate a vaccine against. We optimised that genetic sequence, inserted it into our DNA plasmid backbone, which you can see there as a circular piece of DNA. That's then manufactured, formulated with a little bit of salt and water, and then introduced into subject's skin. And here you can see one of the, and I'm just going to turn on my laser pointer, and you can see here we deliver our vaccine to the patient's skin using our very interesting smart delivery technology device. It's this device here that I'm circling. It's about the size of an electric toothbrush, and I'll talk a little bit more about the details of that. But that really very efficiently introduces the DNA into your cells, allows your cells really to kind of trick your cells essentially into becoming a factory for that antigen of interest, that spike protein, which then produces those antigens and then is able to elicit those downstream effects that I talked about, which are both antibodies and T cells that are able to fight infected cells. So a little bit more about the device, because that's really an area which is different from maybe some other vaccines that everybody's familiar with. So DNA itself can be injected nakedly into the body. However, it's a very inefficient process of getting your cells to uptake the DNA. We've really spent many, many years designing delivery technology to ensure that the DNA is very efficiently taken into the cells. And really what you're looking at here is the result of many, many years of development of a device. And very thankfully, we were developing a device for use in a pandemic setting well before we even knew what COVID-19 was. And so this device has been developed very specifically with a mind to being shipped globally, being used in a variety of different settings, from outdoor vaccination campaigns in Africa to indoor clinics in the developed world. So a very different profile. It's can be used in different humidity settings, different dust settings, different temperature settings. So it's been very specifically designed for that process. And the reason that we use enhanced delivery is really that it results in incredibly improved immune responses and is able to generate that broad immune profile that we believe is necessary really to protect you from these viral pathogens that we're talking about today. The device itself has been evaluated in over 2,000 subjects and has been used for over 12 years. So the device, as I said here, is easy to use. It's a single button press from the perspective of the clinician. You hold it onto the patient's skin, you press the button, and the administration takes really only just a few seconds. And then just to give you a visual of the benefit of enhanced delivery using our device, what you're looking at here on the bottom right-hand side of the slide is rabbit muscle. And on the top panel, you can see we've just injected DNA expressing green fluorescent protein. So really, basically, it's just a marker for where the DNA is expressed. And you can see on this top panel, really, there's very little to no expression of green fluorescent protein. However, when we deliver that with our enhanced delivery technology, you can see the muscles themselves glow incredibly green. And that's really a very nice visual of us tricking, essentially, the tissue into becoming a factory for our antigen of interest. This is a little sort of schematic of how the vaccination process would go. You deliver a standard intradermal injection. It's probably one of the most common injections given in the world because, of course, PPD, tuberculin tests are given this way. So you deliver the vaccine to the skin. You can see a little raised wheel on the surface of the skin. You then apply the device to that raised wheel. You press the button. You wait for some auditory triggers. You wait for a change in light. Then you remove the device and you dispose of the single-use array. So the whole process is completed in just a few seconds. This is a close-up of the device itself. Again, I'll remind you it's about the size of an electric toothbrush. The disposable array part, which is single use, is this part here. So it's about just a little bit over a centimetre in width. It comes with a protective safety cap. And here you can see the back end of the device. And we've specifically designed it so that it uses rechargeable batteries, which can be powered through standard, through the wall charging. You know, any cell phone charger, any mobile phone charger can be used to charge the device. But also in low resource settings for Africa, for instance, you could use solar power or any other mode of powering to charge up the device. And you'll get about 200 uses of the device out of a single charge. So then let's talk about the regulatory pathways. You can probably imagine that the world, in fact, really had never experienced the push to generate vaccines so quickly. And so there were many huge amounts of effort that had to be overtaken from a regulatory perspective to ensure that vaccine development was being carried out in a safe and ethical way. And so certainly here at Innovo, we've spoken, of course, as a US-based company, primarily with the FDA, but also with the Korean Ministry of Food and Drug, and also the MPA in China, because we're also running global trials there. We're also in discussion with the European Medical Agency, because we're really looking at eventually this being a fully globally used vaccine. So as vaccine development always occurs, although of course COVID-19 was slightly different to how you would standardly do it, you design your vaccine, you test it generally in vitro in cells in culture in a lab, and then you move on to animal studies. You know, last year everything like that was done in parallel when usually it's done in sequence, and I can certainly talk about how we managed to speed things up a little bit more in the question and answer session. But really generally you would start with mice, move on to a larger mammal, and then eventually on up to a primate. Last year, of course, we didn't have the time to do that, so we were running everything in parallel. So it's really quite a different experience than our standard vaccine development timeframe. But suffice to say, we were able to use our preclinical animal models to really demonstrate excellent immunogenicity, the presence of those important neutralizing antibodies, and probably most importantly, since it's a surrogate for the efficacy of your vaccines in humans, we were able to show protection in primate and mice challenge models for SARS-CoV-2. So that's really very, very crucial data to have as we move forward. And this data has all been published in a variety of different settings, but probably the most comprehensive is in Nature Communications. So as soon as we confirmed that the vaccine was immunogenic in animals, we of course immediately moved to phase 1 clinical testing. That started here in the US. And just to give you a sense, from the day that we received the viral sequence from China, 83 days later, we were treating our first subject in the clinic. And certainly for us at Innovo, that was a huge record, a massive milestone. We've never moved that quickly prior to COVID-19. Our phase 1 study was our first in human of this vaccine, started in April of last year, and initially started in a small cohort of 18 to 50 healthy subjects. Of course, though, as we all know, older subjects are disproportionately affected by COVID-19, so we immediately then expanded that trial into 51 to 64-year-olds, and then eventually into 65 and older down groups of individuals. We assessed 3 different doses, 0.5 milligrams, 1 milligram, and 2 milligrams, and we dosed each subject twice, so at day 0 and at day 28. We essentially mirrored exactly those same studies in South Korea and China, and those studies are ongoing as we speak. We are just about to finish our phase 2 study in the US, and phase 2 studies are ongoing in Korea and China. And we are looking to initiate our phase 3 efficacy studies in the US and globally later this year. So that's a very exciting time for the company. This is kind of an overview of the clinical design here. You can see we started with a relatively few number of people, and then as we tested the safety, established the safety, then we scaled that up to 120 people in our phase 1 clinical trial. So the purpose of any phase 1 trial really is to assess the safety, number one, of course, and also the immunogenicity. And we did that at different doses and in different age groups within the population. And this is really this is a graph depicting the safety profile of our vaccine. And this is really very important, I think, for those perhaps that have been lucky enough to already receive the vaccines that are currently available, or perhaps have read about those who have. You do tend to get some flu-like symptoms, some fatigue. Certainly I've known a lot of people who have had to spend the day in bed following getting those vaccines. That's not the case with our DNA vaccines at all. As you can see, we have incredibly mild adverse events or localized side effects, as you might call them. Really, it's primarily just localized redness and a little bit of pain following the injection of the vaccines. So we're really very, very excited and enthusiastic about the safety profile of our vaccine, and we believe that it really is very crucial to have safe and effective vaccines. So as important as it is, of course, that our vaccine's safe, it's also important that it generates the right kind of immune responses. And so we, going into the trial, set up really a panel of immune responses that we were going to look at, as we received blood samples from those subjects. We wanted to look at those T-cell responses that we believe are very important, and we also wanted to look at B-cell, those antibody or humoral responses as well. So here's a little bit of the data, and really just in a nutshell, maybe I'll kind of go over at a high level what you see here. And these are our two different of the dose groups, so the 1 mg dose and the 2 mg dose. And you can see, of course, at week 0, the subjects have little to no responses. And then at week 8, post receiving those vaccines, you can see that they have really very nice T-cell responses. And I'll talk a little bit more later on about why we think T-cell responses are so important. And then certainly the same can be said for those binding antibody responses. We're looking both at the spike protein and the receptor binding domain protein here. These are just different elements of the antigen of interest that we've transcribed. And you can really see that at both doses we're getting very nice responses. And then, of course, as you've probably read a lot about, we believe that neutralizing antibodies play an important role in protection of people from SARS-CoV-2 infection. And you can see here that we generate really quite strong neutralizing antibodies in both the dose groups. So this is just, again, a very busy slide, but really just puts together all of the immune data that we received from our subjects in our phase 1 trial. And as I mentioned, we're kind of working through finishing up the phase 2, and that data should be available next month. So then what's the kind of the next step for us? So after phase 1, of course, you move to a phase 2 study, which really essentially is usually just a much larger study and tends to be used to really drill into the dose that's appropriate for a wider range of the population. And that's absolutely what we are doing currently. This is currently just in the US, and we have enrolled 400 subjects. For our phase 3, our efficacy trial, that's going to be a global trial, so we'll definitely have some US sites, but it'll be all over the globe. And we're looking at about 6,000 subjects to be enrolled in that trial, although that would depend very much on the infection rates that we're seeing at that time. So as I mentioned a little bit earlier, in regards to manufacturing of DNA vaccines, it's a process that's actually very easy to do, very scalable. Here in San Diego, we make all of our research DNA here in the lab using fermenters that are about this size, you know, soda bottle size of fermenter. However, of course, when we're thinking of manufacturing this for global usage, the fermenters that we use are essentially, you know, the whole size of a factory floor. They're absolutely enormous. But because the process is so simple, it's very, very easy to scale. And so we've been, you know, working very diligently over the last 12 months to really build up a global presence of manufacturers all over the world so that we can produce and supply vaccines locally, regionally, globally. Yes, it's also a very cost-effective process as well. So let me just move on to here. So as I mentioned, our vaccine will be a 2-dose regime with excellent stability profile. As I mentioned, we've generated preclinical
I think Dr. Kate might have some problem with the connection. Then we advised her to go out from the meeting room and come back. Just let's wait a little bit.
Dr. Kim, can you hear me now?
Yes, we can hear you now. So you have been a little bit disconnected, then you can resume. Thank you.
I'm so sorry about that, everybody. I really do apologize for that. This is really the last section of the presentation today, and I wanted to kind of end the discussion talking a little bit about the threat of the newly emerging strains or variants of COVID-19 that we're seeing currently. I'm sure everybody has read about these or heard about these. I guess it's no surprise probably to anybody that SARS-CoV started to mutate and has done so really quite effectively, unfortunately. And I'll kind of draw your attention to the panel on the top right-hand side. Certainly here in the US, in February, only 1% of the COVID cases were caused by the UK strain, the B.1.1.7 strain of COVID-19. However, now in March, mid-March, it's now the dominant strain in the US. And so it really is a testament to how fast this incredibly infectious variant of COVID-19 has spread, and it's certainly spread globally. It's not just, of course, in Europe and the US. It's I believe, been detected in almost all countries in the world now. You'll almost certainly have heard about the UK variant because it's really been spreading very fiercely. But of course, there's huge concern also about the South African variants and the Brazilian variants. And certainly I was just reading this morning that unfortunately in Brazil, the situation really is very, very dire. Their healthcare systems are almost completely overwhelmed by this new strain of the virus, which seems to have higher morbidity and mortality rates, as well as being far more infectious. So really, we see this as a huge new threat. And the reason I say a new threat is because really so far, it doesn't look like the vaccines that were generated last year are as effective against these new variants of the virus as they were against the original Wuhan strain of the virus. So this is really very concerning and something that the global community has to take very, very seriously. So certainly from our perspective, we were focused originally, a year or so ago about dealing with the pandemic in the US, and that was why we ran our trials there. However, now as we kind of understand maybe more about the epidemiology of SARS-CoV-2, it's looking like it's going to be a more endemic situation just than a pandemic situation. And so we really need to think of it more in a influenza-like situation as opposed to a MERS or a SARS setting. And so really we need to now be thinking about how to deal with the future of this disease, and does that mean annual immunizations? Does that mean seasonal immunizations? And as such, will we need to change the vaccine design on an annual basis to deal with those variants? And as I just mentioned, of course, we're very aware of the UK variant, the Brazilian variant, the South African variant, but really the virus is going to continue to mutate. So we need to be thinking not only just about tackling those new strains of SARS-CoV-2, but also the new strains that will almost certainly start to appear in the coming weeks and months. And so here at Innovo, we have designed what we call a pan-COVID vaccine. So a vaccine that we believe will cover the known variants currently, so that's still the Wuhan, the original strain, but then also these different, the P1 mutation from Brazil, the South African variant, the UK variant, combining all of those into a single vaccine, and also adding a predictive element to it that hopefully will be able to have cross protection to the strains that we don't know about at the moment. And really, just as I think I mentioned in my early slides, as a virus mutates, the nature of an antibody is it's a very specific event. It's very like a lock and a key. A tiny change to a lock or a tiny change to a key, and that interaction will no longer work anymore. And that's very similar between a virus and an antibody. It's a very specific interaction. So antibodies are very, very, very impacted by the mutations within a virus. T cells, on the other hand, are less impacted by those small changes within a genetic sequence. So we believe that vaccines that generate both an antibody and a T cell response are going to be really key in managing the impact or mitigating the risk of the impact of these variants. And I'm just going to leave it there and say thank you so much for your time, for coming today and listening, and I'll be more than happy to take any questions that you might have.
Thank you, Dr. Broderick. It was a very interesting and then stimulating research report from the company which is specifically developing DNA vaccine. So, so now we will take some questions from the audience. So if you have any questions, and then as you indicated, please type on the Q&A. But we have some questions. We can start with the first question. So this question is obviously from me. So there are many companies like AstraZeneca, Johnson & Johnson, Pfizer, Moderna, and they are already tried to commercialize their vaccine being immunized to the world population. Compared to these companies, biopharmaceutical is lagging a little bit behind. So what is the main cause that clinical trial 3 is not started yet, compared to that clinical trial 2 has been ended already?
Yeah, no, absolutely. And we fully recognize that, there's been a huge amount of progress in those kind of what we call the wave 1 vaccines. And, we'll completely admit that, we are behind those wave of vaccines. But the one thing I would definitely say, Dr. Kim, is that with 8 billion people on the planet, we are going to need more than just those handful of approved vaccines. And really, as I tried to kind of mention in my slides, there are certain characteristics about our DNA vaccines that make them really quite appropriate for different global settings and for different global populations. So we absolutely believe
Sorry, Dr. Kate, are you still there? So you were cut off. So let's wait a little bit because Dr. Kate has the same problem during the presentation. She's disconnected somehow. Dr. Kate, do you hear me? So your mouse cursor is visible on the screen, but we cannot hear from you anything now. So I suggest you come out of the meeting room and come back.
Can you hear me now, Dr. Kim?
Yeah, now we hear you.
Yeah,
So
I'm
you are coming back.
so sorry about that, everybody. That's terrible. I'm really sorry. So I don't know, Dr. Kim, where I got cut off in my answer to your question, but suffice to say, I think there really is. There's a lot of room for different vaccines with different profiles, and I think with the threat of the emerging variants, specifically DNA vaccines really meet a lot of the criteria that are required to mitigate that risk as something that would be needed as an annual vaccination. So question number 2 looks like, what is the difference between selectra and normal syringe to deliver vaccine under the skin? So yeah, that's a great question. So a normal intradermal injection is using a very thin gauge needle. It's using an insulin needle actually, so it's a 32 gauge needle, and it's really just inserted in a very shallow under the surface of the skin. So we perform our injections that way. Selectra device, though, the delivery device, has 3 very thin gauge electrodes. They're about 3 millimeters in length, and they're penetrated into the skin where the person's received the vaccine, and it's left in there to deliver those electrical pulses that ensure that efficient uptake of the vaccine.
Thank you, but you cannot use Selectra for other vaccine from other company.
Not from other companies, no, but it's used for all of our DNA vaccine deliveries.
Okay, thank you. Now, further questions?
So how much to get funded by government for your DNA to facilitate its delivery to world? Yes, so we are incredibly honoured to have been supported pretty extensively by the US Department of Defence here in the US to support our COVID vaccine development. We're also very honoured to have been supported by the Bill and Melinda Gates Foundation and also CEPI, which is the Coalition of Epidemic Preparedness. So we've had a lot of supporters who have financially assisted the efforts of this development.
Thank you. So next question. So this is from Dr. Julie, I guess. So this is almost similar like my first questions. And compared to traditional injection of the vaccine, and then using selectra, so how do you see the feasibility of deploying your vaccine to the masses?
Yeah, and I think that's a really important point. And I guess I think that it's an extremely easy to use device. It's specifically designed to be very easy to ship, to stockpile, to store. And I guess when you contrast a device that's, this big versus a -80 freezer that can take up a small room, I think using an injection device is actually very, very simple and very easy to include into the mass vaccination campaigns. And we're using it pretty extensively in Africa for different vaccination campaigns. So I don't see it as an issue for rolling out for global usage, and actually see it as a far simpler addition over the needs for ultra cold storage, which some of the other vaccines require.
Okay, thank you, Dr. Kate. And another question from Dr. Lawrence Stanton. So as you know, most of the vaccine from different company are composed of 2 shots. So there is any way to get done by 1 shot? Why with 2 shots?
Yeah, that's a great question. So at the moment, the only vaccine that's currently being used that's a single shot is the Johnson & Johnson vaccine. So that's a huge benefit, as you can imagine, that only a single shot. For us, Moderna, Pfizer, the AstraZeneca Oxford team, we all require 2 shots to generate that. So you certainly will get some protection after a single shot, but to reach those high levels of efficacy, for Moderna and Pfizer, which are up in the mid-90%, which is absolutely remarkable, you do need the 2 shots. You just have to have that to prime the immune response.
Okay, thank you. So here on the monitor, there is question 5. So regarding the selectra used for your product, Innovo 4800, so why is it taking so long to get back to the FDA about this selectra device? My understanding is the selectra device has been already approved.
So it's not an approved device, but we've used it extensively in our other phase 3 trials, in all of our clinical testing all over the globe. So yep, the FDA had some very specific questions about the utilization of the device, and we're just working through generating those packages, which will be ready in time for our phase 3 start, with the concurrence, of course, of the FDA.
Thank you. I have difficulty to read the question from Q&A because I cannot expand the here space here. So somebody who can see questions more clearly, Omar, you can see question. I can read only 1 or 2 sentences here from here monitor. It's very difficult to see whole question because of this big blank boxes here. So one question is saying, okay, so 6, could you please evaluate in 2 doses? This was already answered by Dr. Stanton. And another question here is, okay, can you modify your applicator to be the only device you need instead of the current 2-step?
Great, absolutely fantastic question. And yes, we're working on that currently. It won't be available for the trials in the near future, but that's something we definitely want to do, is combine the injection and the electroporation into the same device.
Okay, thank you. So, okay, next question is whether your DNA vaccine is more versatile to adapt to new variant with more severe effect. So without having go through the same study and approve process again. So it is similar to flu vaccine development, do you think?
Great question. And as you can imagine, the regulatory authorities, the FDA here in the US and all the other global regulatory authorities, are really trying to wrap their heads around at the moment. You know, what is the impact of these new variants, especially on the basis that the vaccines that are currently being used, we know are not as effective against the variants as they were against the original strain. So one of the great things about our DNA technology at Innovo is that we can modify the genetic sequence very, very quickly. So that can be done, you know, in a very short period of time. So as I mentioned, and I think in my last slide there, we're currently working on a pan coronavirus, pan COVID-19, sorry, vaccine that will cover the known variants and also hopefully have some predictive power over the unknown variants at the moment. It will have to go through, we believe, clinical testing. Of course, it is a new product, but potentially there may be paths to kind of use some of the learnings that we have from our original vaccine and apply those to the new designs on the basis that the designs really aren't terribly different from one another. However, that's not our decision. That would be, of course, the decision of the regulatory authorities.
Thank you. So, okay, next questions. Considering the regulators have allowed the streamlined process for vaccine development and policy has been successful, that the current process will become the norm or at the forefront of the research, do you think that some of the administrative process to approve all of the clinical trials should be better than now?
It's a great question, and you know it's a question that I get very frequently. Is well, now that you know, science has shown that we can develop vaccines in less than a year, is that now going to be the new normal? I don't think so. I think, you know, the whole scientific community was geared up and really genuinely worked 24/7 for the last year to accomplish what was accomplished. I don't think that's necessarily sustainable. And I certainly think from the regulatory authorities, I don't think they could continue to work as fast as they have been. I think that would be an awful lot to ask of everybody involved. However, I think there certainly has been lessons learned in areas where we could potentially have packages out to the regulatory authority in advance, so that they would have been approved in advance and it wouldn't have to be all done kind of as a reactive mechanism. So I think there are a lot of areas where we will see some acceleration in the future, but I don't know that we as society should necessarily expect that kind of rolling out vaccines will now be as fast as it was for COVID. I think that probably isn't very likely.
Thank you, Dr. Kate. So next question. Since it's a DNA-based vaccine, I'd like to know if you consider what's the effect during the preclinical studies, because you have already conducted clinical trial 1 and 2. There was any kind of the side effect and then potentially leading to burst effect, do you think?
That's something obviously that we take very, very seriously. And over the 20+ years that DNA vaccines have been used, it's been very clear that the DNA plasmids don't and can't integrate into the human genome at all. So there's no evidence of integration events. And in fact, we've shown that now explicitly through reproductive studies in animal models. So that's something we feel. So there are no associated birth defects with DNA vaccines. In fact, the side effect profile from DNA vaccines is incredibly mild.
Thank you. So there was some fear among the population about this possible side effect for the potential of the DNA vaccinated being integrated in the human genome. Then thank you for a clear answer from Dr. Kate. So next question, is DNA-based vaccine? I'd like to know, no, this has been done. And there is other question now. Okay, so I have one more. So there are people attended for clinical trials 1 and 2, So did you follow those patients who attended in clinical trial and 1 and 2, whether they have developed the additional symptom of the COVID-19? I mean, they are newly affected by COVID-19 or their effect has been continuously immune to any kind of the COVID-19 with a different variation?
So we certainly are continuing to follow all of our subjects enrolled in our trial. It was kind of part of the design of the trial, was to continue to follow those subjects, and that'll certainly be data that will be released as we kind of complete that analysis.
Okay. So I think all of the question has been nicely answered and clearly replied. So now we are at the end stage, and it was really great to be updated of the new development of DNA vaccine from the forefront company of the Innovo Pharmaceuticals from San Diego and Pennsylvania. So Dr. Kate Broderick, thank you so much for taking your time to update us this nice development. We hope to hear soon from new development and then commercialization of your vaccine into the world population. So thank you for attending. So it's evening time. So have a great evening for all of this audience. So see you soon in next month. Thank you.
Thanks.